Hello and welcome. In this lesson, we're going to look at water relations in plant cells and specifically to demonstrate osmosis using plant tissues. Now, the plant tissue that we are going to use is derived from the potato tuber. This is an Irish potato tuber, which is basically a storage organ found in this particular plant. Now, using a scalpel, we make 3 cubes of about this size, and we bore out cavities at the center. So there'll be a hollow cavity at the center of each of the 3 cubes. That is, you can do that using a scalpel or a sharp knife to prepare a cube that is obtained from the potato tuber, and then we dig out this hollow cavity at the center. Then each of the 3 potato tuber is treated as follows. The first one is given the A treatment, in which salt crystals are placed at the center of the cavity, and the tuber is left unboiled, then placed in a petri dish that contains distilled water. The second tuber is given the B treatment, in which it is left unboiled and placed in a petri dish containing distilled water, but without any salt crystals at the center. And then the third tuber is given the C treatment, in which first it is boiled for about 5 to 6 minutes, and then salt crystals are placed at the center of the hollow cavity. Then the tuber is placed in a petri dish containing distilled water. The 3 tubers are left to stand for about 30 minutes, after which the following observations are made. In the first tuber, it will be observed that water will accumulate in the cavity, and the level of the water in the petri dish is going to go down. In the second tuber, we will see that no solution is formed in the cavity, but the level of the water in the petri dish reduces slightly. While in the third cube, we'll see that the level of the water in the petri dish remains the same, and the salt crystals remain unchanged, that is, they do not go into solution. Now, explanation of these observations. Now you can see that in the first potato tuber, the salt here in the cavity dissolves in the thin film of water found on the potato tissue, thereby forming a concentrated solution here at the center. This concentrated solution is hypertonic to the potato cells lining the cavity. So what happens is that water moves by osmosis from these cells lining the cavity into the cavity, thereby increasing the level of the solution there. The cells lining the cavity become hypertonic to the inner cells. Thus, water moves by osmosis from the inner cells to the cells lining the cavity, and this goes on until the outermost cells draw in water from the petri dish by osmosis, leading to a drop in the water level of the petri dish. So that's why you see after 30 minutes, the solution level in the cavity increases because water is drawn from the cells in the potato tuber by osmosis, and in turn, they draw water from the petri dish by osmosis, replace the amount that is lost to the cavity, leading to a drop in the level of water in the petri dish. In the second tuber, we see that no solution forms in the cavity, but the water level reduces slightly in the petri dish. Now, how is that? This comes about because the cells in the potato tuber are hypertonic to the distilled water in the petri dish. So, by osmosis, water molecules are drawn from the petri dish and into the potato tuber. And this is what brings about a slight drop in the level of the water in the petri dish. In the third tuber, where there is no observable change, comes about because boiling destroys the cell membrane. Now, with the destruction of the cell membrane, no osmosis can take place. So the salt crystals remain undissolved. Water in the petri dish cannot be taken up by osmosis because of the destruction of the cell membranes of the cells in the potato tuber. So consequently, no water is taken up into the tuber, and there is no dissolution of salts crystals in the potato tuber.